Okay, probably we should start with this symposium number 4 that will focus on neuroimaging methods and with more clinical kind of uh, lens. So it's looking more like potential clinical application in neuroimaging, but also EEG methods as well. We have 3 speakers, including myself. I'm supposed to be sharing this session, but I can see Stefan, so I'm not sure if Stefan will join at some point. Well, this is the last speaker, but I'm delighted to see that Thomas is already here. So Thomas, are you okay to share your slides and go ahead with your presentation?
Sure. Can you see my slides?
Yeah, perfectly well. Thank you very much.
Okay. Yeah. Hi. I think I would just get started. So first of all, I assume most of you actually work on EEG. I have to confess that most of the stuff I'm showing you is actually fMRI, although that some of the stuff is actually applicable to EEG, as I will point out. So basically today I'll talk about how we can jointly optimize sample size and scan duration to improve phenotypic prediction using functional MRI. By the way, oh yeah, I've muted myself just okay. So many of you might have seen this study from Scott Marek showing that brain-wide association studies with brain MRI requires thousands of individuals to be reproducible. However, reproducibility is not the same as validity because a broken clock is highly reliable, but it is only correct 2 times a day. So I'm actually more interested in individual level prediction, which is of course a big topic in fMRI. Many studies have used function connectivity to predict brain maturity and fluid intelligence at the individual level. And the hope is that we can eventually use function connectivity to predict disease progression or to select best treatment for an individual with neurological or psychiatric disorders. One of the bottleneck in the field is that we would like to improve prediction accuracy. And it is well known from machine learning that we can simply increase sample size to improve prediction accuracy. So here's a UK biobank experiment. My student He Tong trained different models to predict fluid intelligence. Here we consider kernel regression and 3 deep learning models. So y-axis is the prediction performance, the higher the better. X-axis the number of training subjects. As the number of training subjects increases, all regression models keeps improving. What is interesting is that sample size has a much larger effect than the different prediction models. And these results also hold true for other phenotypic measures. So this is coming from an engineering background. This is really sad for my students who like to come up with these complex models. But actually, just by increasing sample size, we can increase prediction accuracy a lot. What about scan duration? If sample size is fixed and we increase scan duration, prediction accuracy for certain cognitive measures also improves. So here's a result from Professor Jing Wei from Beijing Normal University. X-axis, the number of fMRI time points. Y-axis, the prediction accuracy for the picture vocabulary tests in the Human Connectome project. As we increase scan duration per participant, prediction accuracy actually increases, as we expect. But we still do not know what is more important, sample size or scan duration. So this is work done by Leon, Shaba, and Shaoshi from my group. You can check out our preprint here. So let's start by looking at the cognitive factor score in the Human Connectome project. So brighter color means better prediction accuracy. X-axis is a scan duration for each participant, and y-axis is the number of participants. For a fixed scan duration, so let's say 34 minutes, right? The prediction performance increases with more participants. This is consistent with the UK Biobank results I've shown you. On the other hand, if we fix the number of participants, the prediction performance increases with greater scan duration per participant. Again, that's not surprising. And here's a full plot. So along a black isocontour line, the prediction performance is constant, although the scan duration and sample size are changing. So essentially, we can compensate for shorter scan duration with a larger sample size. And conversely, we can compensate for shorter for a shorter, we can compensate for a smaller sample size with greater scan duration. But even from this plot, we know we can trade off the two parameters, but we still do not know what is more important. What is interesting is that empirically, we find that scan duration and sample size are broadly interchangeable. So in this plot, y-axis is the prediction accuracy of the cognitive factor score. So this is in the ABCD study. X-axis is the total scan time. So this is defined as the number of participants multiplied by the scan duration per participant. And amazingly, the total scan duration explains prediction accuracy really well. So here, each color represents different training set size. That color corresponds to larger training set size. If the dots of the same color represent different scan duration. And looking at this data, you can see that scan duration explains prediction accuracy really well. And by staring at it, we thought that, hey, maybe the logarithmic function might explain these dots really well. And so here's what I'm showing you here. So in this plot, the x-axis is now the log of total scan time, and the y-axis is the normalized prediction accuracy. So different color corresponds to different phenotypes. We see that across different phenotypic domains, there's a linear relationship between normalized prediction accuracy and log of total scan time. This relationship holds for up to 20 to 30 minutes of fMRI scan time. Beyond that, we start to see a plateau in the prediction accuracy. So here's the cognitive factor score in the Human Connectome project. X-axis is the total scan time. Y-axis is the prediction accuracy. We see that total scan duration explains prediction accuracy really well, except that the accuracy plateau and maybe even slightly drop after 20 to 30 minutes of data. So overall, it seems that scan time and sample size are interchangeable up to 20 to 30 minutes of fMRI data. But there's diminishing returns for scan time relative to sample sizes for longer scan time. So how can we explain this? Luckily for us, Tom Nichols, who is a statistics expert, helped us derive a closed form formula for prediction accuracy as a function of sample size N and scan time per participant T. K0 is the maximum possible prediction accuracy for the phenotype. Obviously, that will vary according to the strength of the relationship between function connectivity and the phenotype. And there's a term containing just sample size N, and there's a second term NT, which is total scan duration, sample size N times scan duration T per participant. And we can, if you look at this, as we increase N and T to infinity, this square root term goes to 1, and we achieve the maximum possible accuracy K0. So let me provide some intuition behind Tom's derivation. Intuitively, we can decompose prediction into two parts. One part relating to an average prediction, which is common to all participants, and the second part relating to an individual's deviation from this average prediction. The uncertainty of the first part, which is the average prediction, scales with 1 over n, like the basic standard error we see in statistics. It does not depend on t because this is the average prediction. The second part is the individual's deviation from the average prediction. And in the case of linear model, right, we can think of this part as the function connectivity of an individual multiplied by the linear regression coefficient. Now, the uncertainty of function connectivity scales with 1 over t, while the regression coefficient scales with 1 over n. So if we multiply 1 over t with 1 over n, we get 1 over nt, which is the uncertainty of the second part. And so now the first part contributes 1 over n, and the second part contributes 1 over nt, which kind of explain the formula that Tom's derived. This is an oversimplification, right? We make certain assumptions. For example, we assume that the fMRI time course is stationary. So this means that the statistics are the same at the beginning of the scan and at the end of the scan, which might not be true. The derivation also does not tell us the relative importance of the 1 over n term and the 1 over nt term. So what we can do is that there are 3 unknowns, K0, K1, K2. We can fit this to real prediction accuracy data. And the goal here is to determine whether our theoretical model can explain the empirical results even though we have these simplifying assumptions. And also, we want to determine the relative importance of 1 over n and 1 over nt. And the good news is that this model can explain empirical prediction accuracy really well. Here, I'm showing you an example phenotype in Human Connectome Project, spatial orientation. The blue curves corresponds to Tom's theoretical model. Basically, when the fMRI scan time is short, the 1 over nt terms, so this 1 over nt terms dominates the 1 over n term, right? So scan time and sample size are roughly interchangeable. Now, because of the 1 over n term, right? So as scan time becomes big, this 1 over n term starts to win over the 1 over nt term. So eventually, sample size becomes more and more important relative to scan duration. Now, does this mean that we should always maximize sample size over scan time, right? I'm telling you that sample size and scan time are interchangeable. And in fact, sample size is slightly more important than scan time. Should we scan our participants for only 2 minutes and then go for the largest sample size? Now, intuitively, you'll probably think, right, we obviously should not do this, because why would we go through the trouble of bringing in our subject in and only scan our subject for only 2 minutes? That seems like a suboptimal thing to do. And indeed, this intuition would be correct, right? We can encode this intuition in terms of an overhead cost associated with each participant. This overhead cost might include recruitment cost. It might include non-imaging costs, for example, blood markers or cognitive tests. It might include non-fMRI imaging costs, for example, PET data or EEG and so on. And so what we can do is that we can use Tom's formula to inform study design while accounting for this overhead cost. And to do this, we can think of the square root term as a fraction of maximum accuracy, which we can average across phenotypes and extrapolate to different sample size n and scan duration t. So for this, to create these resource for future study design, we consider 6 data sets. We found that 76 phenotypes across phenotypic domains have a good fit to Tom's model. Our data consists of children, young adult, elderly participants from China, Singapore, North America. We have healthy participants, as well as participants with psychiatric disorders, including major depression and other psychiatric disorders. We also have participants with AD dementia and mild cognitive impairment. And our acquisition is diverse. It ranges from single band, multi-band, multi-echo, multi-band. Our data consists of scanners from GE, Philips, Siemens. And we also consider both resting state and task state functional connectivity. And so by averaging across 76 phenotypes, right, across the 6 data set, we get this plot, right? This is the plot of maximum accuracy as a function of the number of participants and the scan time per participants. So as we go along this black isocontour line from left to right, the fraction of maximum accuracy is constant, right? As we go from left to right along the black isocontour line, the total scan duration actually goes up because sample size is more important than scan time. So as we go from left to right and we increase the scan time per participant, we can decrease sample size, but not so much. But remember, we have the overhead cost associated with each participant, right? So even though the total scan time is going up, but the cost might not go up. In fact, the cost might go down. So for example, let's consider a small study. So let's say the total fMRI budget is $100,000 and the scan fee is $500 per hour, right? So y-axis here is a fraction of maximum accuracy. X-axis is a scan time per participant. I'm going to show you 4 curves corresponding to different overhead costs per participant. And here are the 4 curves. As we go from left to right, the scan time per participant increases, but the sample size decreases, so that the fMRI budget is held constant at $100,000. The solid circle, these solid circles here show the optimal scan time per participant in order to maximize prediction accuracy. We also provide an error bar. The error bar shows the range of scan time for which we can achieve within 1% of the maximum prediction accuracy. Notice that the error bar is short on the left side. The left side is short, but the right side is long. And that's because the curves rise sharply when scan time is short before slowly decreasing as scan time increases. So when overhead cost per participant is, let's say, $500, let's say this is the blue curve, then the maximum prediction accuracy is obtained with 119 participants and 67 minutes of fMRI per participant. Now, if we relax the threshold a bit, if we only consider the 1% maximum accuracy, so at the minimum, we need to collect at least 25 minutes of fMRI, which is much longer than what most studies collect. Let us now consider a large-scale study, right, where the total fMRI budget is 10 million. So this is likely a multi-site study where data is collected from multiple MRI scanners, right? The first thing we notice as we go back and forth is that the fraction of maximum accuracy is much higher when the fMRI budget is higher. And this makes sense because when our budget is higher, we can increase sample size and scan duration. And as we go from left to right along each curve, the scan time per participant increases, but the sample size decreases. So the total fMRI budget is held constant at 10 million. In this case, when the overhead cost, let's say we varies it from $500 to $5,000, right? The minimum scan time goes from 8 minutes to 42 minutes. So for very large scale studies, which might have a high overhead cost because of the multi-site data collection, the minimum scan time can vary quite a lot. I'll move on to some robustness analysis, right? Here's a medium scale study. So here fMRI budget is $1 million, right? So in this plot, we assume the MRI costs $500 per hour and the overhead cost is $500 per participant. The plot is the same as before, but now each color represents one data set. We see that the 1% scan time ranges, the error bars, are highly overlapping across data sets, suggesting robustness across data sets. Right? We can repeat with different phenotypes. Here are 7 phenotypic domains. The brackets shows the number of phenotypes in domain. So cognition has 39 phenotypes, while emotion has 1 phenotype. Again, we see that the optimal scan time ranges are highly overlapping across phenotypic domains. The emotion domain is an outlier, but there's only 1 phenotype in that domain. So I don't think we should overinterpret this. Our results are also robust across resting state and task state functional connectivity. And in fact, we obtain similar results if we change the number of parcels or if we look at cortical-subcortical connectivity. Results are also robust if we split the data collection into 2 sessions. So in the case of the human connectome projects, participants are scanned on 2 separate days. And so we can simulate what happens if we scan participants on 2 separate days instead of 1 day. To summarize, we find that the total scan duration explains individual level prediction accuracy via a logarithmic model. And the implication is that sample size and scan duration are broadly interchangeable up to 20 to 30 minutes of data. The returns of scan duration diminishes relative to sample size, which we can explain with a theoretical model. This theoretical model is very general, and we can actually apply it to EEG data if you're interested in applying it, right? Or any time series data where the uncertainty of the measurement decreases with 1 over t. In fact, it doesn't even need to be functional connectivity. If you look at task evoke activity from EEG or from fMRI, right? As long as the uncertainty of the measurement decreases with 1 over t, we can use Tom Nichols' theoretical model. When considering fixed cost per participant, small-scale studies benefit from longer scan duration than typically assumed. Even large-scale studies might benefit from longer scan duration if the overhead cost is high. And this may be likely because for a very large-scale study, you have multi-site data collection. So we have these additional overhead costs associated with additional participants. Our results are robust across phenotypic domains, across age group, across racial group, across health status, whether it's healthy participants or participants with psychiatric disorders or Alzheimer's disease, is robust across MRI, functional MRI acquisition, is robust across resting and task state function connectivity. More broadly, our results argue against traditional power analysis, where our only inputs are sample size. These kind of power analysis inevitably point towards maximizing sample size while minimizing scan duration because of the limited budget. So the resulting prediction accuracy are likely lower than what can be produced with alternate design, which impedes scientific discovery. So we will be making our empirically informed reference available for future study design. I'd like to thank all my funding sources and support. I'd like to thank my lab members and collaborators. Thank you very much.
Thank you very much, Dr. Thomas, for this very interesting and useful kind of presentation with very important practical implication. I have a few questions. Hopefully, we will have some time to share at the end of this presentation. So I am next, and I will change gear a little bit, and I will share my screen. Please let me know when you see my screen. Can you see my slides?
Yes, we can see it, Professor.
Brilliant. So I'll change gear a bit. I will be sharing with you some recent insights about studies where we are combining new cognitive marker with EEG marker or EEG recordings as well. A bit of neuroimaging, not too much, but it's mainly focused on the potential usefulness of combining cognitive and EEG assessments in the potential identification of these transition points that we are learning about as appear to along the continuum from normal aging to Alzheimer's disease, dementia. So we know that from the EEG point of view, we have faced long-standing challenges, meaning in clinical settings. And one of them is because we know well that despite EEG is considered a kind of sensitive assessment tool, and we have heard quite a lot about the EEG as a tool for mechanistic rather than diagnostic approaches, but we believe that it holds potential for the two. However, what we have seen so far in clinical setting is that it's very, very unlikely to find any pathognomonic characteristic feature of EEG that can suggest the presence of any type of dementia. We see general feature like a slowing of fast rhythm and activity, increase of slow frequency activity, but that's not specific to dementia. This can be observed in many neuropsychiatric disorders. So we have seen also the increasing of use of resting state EEG. So I would say that the literature is very tilted towards a study reporting on EEG collecting during resting state for a number of reasons. From a practical point of view, it's the most commonly type of recording that we see we gather in clinical setting. It's easy to share, it's easy to analyze, and also is cost effective because it's not too costly time-wise, nor from a cognitive point of view when we are studying people who are experiencing cognitive decline. And however, the challenge with these emerging trends, emerging developments in resting state EEG is that they are sensitive. They seem to be able to pick up changes and classify patients from control with relatively good level of accuracy in terms of sensitivity, but their specificity is still lacking. So they fail to tell us, as Professor Paulo Orsini mentioned earlier, what kind of condition, what kind of disease. And that's why he remains skeptical about the potential diagnostic value of the EEG. However, we believe that this notion is changing rapidly with the advancements in mathematical AI and other procedures that are unveiling the characteristic of the EEG as a standalone tool or as a tool combined with other methods in an unprecedented manner. So we know that this journey from normal to pathological aging is a long process that starts decades before people develop symptoms. It's a very long silent period, and then people start gradually losing their cognitive and functional abilities. We know that in the area of cognitive assessment, we are facing great challenges because we are kind of lacking tools that are sensitive to the very early preclinical stages of these neurodegenerative diseases. And because prevention is the ultimate goal at the moment, so we do need assessment tool that can help us identify the earliest potential neurocognitive deficit that can be associated with the risk of developing dementia in the future. And we also need to learn from an electrophysiological or neurobiological point of view, what are the underpinnings of these cognitive abilities so that we can develop biomarkers that can anticipate the diagnosis. We have been working on that front for quite a while now. My team developed a memory paradigm. It's a simple task, and as I said, I will mainly focus on task-related brain activity, not much on resting activity, to make the case for why this could be a potential pathway to improve the detection of these transition points. So this memory binding task that was recommended by the Joint Program for Neurodegenerative Diseases in Europe is a simple short-term memory task that assesses the ability to remember over a short period of time shapes, colors, or combination of shape and color. So we ask people to remember shape and then to decide if the shapes that follow are the same or different, remember color, and then decide if the color is the same or different, and remember shape and color together, combination, and then decide if the combination are different. This task has now been recommended by this consensus group because it seems to be very sensitive to the preclinical stages of Alzheimer's disease. So what we have been doing is investigating the electrophysiological neural correlates of this memory binding ability that appears to be informing about the very early stages of the transition from normal aging to Alzheimer's disease. Because in addition to being sensitive to Alzheimer's disease, it has also proved to be specific for this particular type of dementia. In this study, for example, in collaboration with the group of Antioquia in Colombia, Professor Francisco Lopera and Dr. Jaquel Quiroz from Massachusetts General Hospital, we explore using amyloid and tau PETs, the association between this memory binding ability and the accumulation of these abnormal proteins in the brain in carriers, in mutation carriers of this autosomal dominant type of familial Alzheimer's disease. And we look at carriers who were asymptomatic, but they were A beta positive, A beta negative, and we also look at carriers that were in the tau pathology stages. And we saw a significant association between amyloid beta accumulation and these memory binding abilities prior to the association with tau pathology in carriers that were still in the asymptomatic stages. And we have now replicated this observation in other samples of sporadic Alzheimer's disease, confirming that this cognitive marker seems to be informing about the very early neuropathological changes that happens in the brain of people who are developing Alzheimer's disease. So this is very reassuring because we can now move forward after this validation and explore what the EEG can tell us. So the case I'm trying to make here is the one that using EEG drone recorded during task performance, we can identify changes, physiological changes that underpin this particular ability from normal aging all the way up to dementia of Alzheimer's type. So we have seen that in healthy aging, there are significant changes characterized by reorganization of brain activity. There are a lot of compensatory changes happening in older brains in order to cope with the aging process and keep a cognitive level high. So we know that those mechanisms in normal aging start declining in people who are departing from normal aging trajectory towards pathological aging. And at some point, these physiological compensatory changes continue, but they fail to support behavior. So we see a dissociation whereby people progressing from normal aging to dementia tend to recruit more activity. However, their behavior keeps dropping gradually as the disease progresses, up to the point where there is no more room, no more neural resources for either physiological nor behavioral compensation. So then this diagram tries to illustrate this proposal, and I will share with you some data that we have been recording recently that seems to support this notion using EEG. So the idea, very, very briefly, so there are a lot of studies in this slide, but this is just to highlight that knowledge have accumulated supporting this notion that with age, our brain reorganizes itself. As we grow older, there are changes in the way the brain organizes because there are changes in the number and strength of connections between different parts of the brain. And this has been observed in association with high level of cognitive ability, meaning that for older people to retain a high level of cognitive function, they tend to recruit more than they used to in the past. So we have seen that this has led to the proposal of new theories such as the cognitive reserve that explains why some people are more successful in utilizing these brain resources, brain reserve, in order to cope with the with aging changes, and also what are the potential drivers of this cognitive reserve, which are not now being considered as protective factors against dementia. And there are many intervention studies being developed following this evidence. So in this study, this is an MRI study. This is the only MRI study I will show, because in this study was one of the first one that we saw that for people with mild cognitive impairment, so these are older adults who are losing their cognitive abilities and gradually losing their functional abilities, but they are still independent. These people are at a very high risk of progressing to Alzheimer's disease dementia. We assess memory in an incidental emotional memory paradigm. And what we found in this study, in a nutshell, is that behaviorally, this patient with mild cognitive impairment and healthy control show very similar level of performance across the different task conditions. There was a slight drop in MCI, but there wasn't a major dissociation across task condition, meaning identifying emotion versus neutral images. Something that we didn't see in patients with Alzheimer's disease dementia. And when we look at the neuroimaging data using voxel-based morphometry analysis, what we found is that to achieve that level of performance, MCI people needed to recruit more neural resources in the medial temporal lobe, that especially the hippocampus, than the control group did. So that was a very early observation leading to this notion that there seems to be reorganization, functional reorganization of the brain undergoing neurodegeneration in order to cope with these dramatic biological changes. More recently, one of my PhD students used EEG and combining EEG with the memory binding task that I already shared with you. She observed in a group of older people and younger people that even when performance of the older and the younger group was very similar, in fact, in some instances, older people outperformed younger people. It's a high performers or older group. But that was possible because, as you can see in the ERP data, older people consistently over-recruited across pretty much all the brain region that we recorded in this study, indicating that, or at least suggesting that, for them to keep that level of performance high, they needed more resources. And when these resources are available, compensatory changes can happen. And we have now seen that this same task is pretty good at predicting who among the older people can progress to the early stages of Alzheimer's disease. So we call the very early MCI stages. These tasks perform better than other highly recommended memory tasks in the prediction of conversion from normal aging into very early stages of pathological aging that will eventually lead to the development of dementia, indicating that even though there are these compensatory changes happening supporting the function, it holds sensitivity for the very early preclinical stages of the disease. Now, when people move from normal aging to mild cognitive impairment, so this compensation starts failing behaviorally. So what we see now in patients with mild cognitive impairment of the sporadic type, older people with mild cognitive impairment, is that they show a significant deficit in this memory binding ability, and this deficit is accompanied by a significant reduction in neural recruitment in their ability, in these oscillations or ERP activity associated to this task in frontocentral, but also in parietal occipital regions, indicating that this drop in performance is now underpinned by a reduction in the neural resources available. So as I said, compensation seems to be going down the hill at this particular point in time. So this sample that I presented here is a small sample recruited in clinic. However, this is a larger sample of community dwellers. These are it's a community-based study that we carried out in Colombia, in very rural areas of Colombia. And as you can see, it's a pretty good sample size. We had 210 participants, half control, roughly half patient with MCI. And in this group of MCI, so I should mention here that these participants were recruited in the community, so they had not sought medical advice or help at the point of entry to the study. And we invited them through community center, through social media, to enroll in the study. And when we apply the classical criteria, we identified that almost half of them have some level of mild cognitive impairment, but in very, very early stages. So here, what we saw was something quite different to what we have seen in the previous study. Here we saw a combination of increased activity over recruitment. As you can see here, these are areas of the brain where MCI recruited more resources than control when they were performing the memory binding task. And when we compare this over recruitment of brain resources during task performance with, for example, the accuracy of other very well-known memory marker for Alzheimer's disease, the results were very similar. I mean, it's not huge classification accuracy. We're talking about 0.7 around that level. But again, this is very early stages, community-based sample. So it's quite promising to see that this methodology can help us identify people at risk of dementia at this earlier stage. However, I should note that I'm talking about over-recruitment or instead of reduced recruitment or neural resources. So then that's something that we need to keep an eye on. So I will skip this one. So then finally, what we know that when people get to the final stages of the disease, pretty much everything is going down the hill. So we see that there's a loss of a gradual slowing of EEG rhythms. So we see increasing slow activity, this a massive decrease in fast activity, connectivity start disappearing. In fact, this is considered a brain disconnecting syndrome. So we see that brain coherence and other connectivity metrics start failing massively, and recruitment in event-related potential is going down as well, indicating that the biological resources needed for compensation are becoming gradually unavailable. And that what happens in the advanced stages of the disease. So then, in a nutshell, my message, what I'm trying to convey is that we know that there is a long and silent period across the lifespan, and especially there are decades before people develop cognitive decline associated with dementia, and this is known as the preclinical stages of the disease. We call it normal aging, but we know that changes are already happening in the brain that can be detected with other biomarkers. We have been trying to explore if the EEG can also provide some insights. People then move into the prodromal stages, which are these that anticipate and are indicating risk of progressing to dementia. In normal aging, now you see the EEG, there are compensatory changes that allow older people to maintain a high level of function, of cognitive ability, and that will depend on the cognitive reserve. However, we know that those who are living the normal aging trajectory start losing these biological resources gradually. There is still compensation, as you can see here. This is cognitive ability declining and physiological activity trying to compensate, but clearly failing, because at this point in time, this increased EEG activity is no longer leading to improved behaviors up until the point where both functional and cognitive abilities are massively and grossly lost. We are also learning that there are older people who tend to age quite successfully. These are known as the super agers. We are seeing older people who can live well into their 100 years, keeping cognitive ability very high. This is a group of older people who are now being thoroughly investigated, because the number of people living up until that age is growing rapidly across the globe. We are learning that this preserve ability into very late life is also accounted for by functional reorganization. However, we don't have many studies using EEG in this particular area. So in all, there seems to be an interplay between a clinical and electrophysiological threshold, whereby people meeting clinical symptoms are already developing physiological changes that can be detected prior to the diagnostic criteria. That's the I would like to highlight particularly this area here, this area where the clinical threshold has not been reached, but there are significant physiological changes that could potentially inform about not only the risk of developing the condition, but also the progression of the condition, providing a tool not just for prediction and early detection, but also for monitoring disease progression. We have been working together collaboratively across the globe. We have put together this consortium. It's called the EuroLab EEG Consortium. It's now considered the largest EEG database for dementia that there is available in the world. It's still a collaborative project. You are all very welcome to join. If you drop me an email, I will be very happy to provide more information, to help you to sign up, because this is all about sharing. As you have seen for other resources here, EuroLab, the EEG Consortium, is a big database of EEG from people with dementia, of different type of dementia, that we want to make available widely for collaboration, reproducibility, and also to support capacity building as well. So thank you very much for your attention. If you have any question, I'll be happy to take them.
Thank you very much for the invitation to present at the GBC General Assembly on the use of oscillatory and network markers for localization of epileptic foci. So sometimes when I'm tasked to analyze MEG data for pre-surgical evaluation for focus localization, I get data like this. These are about 5 or 6 seconds of resting state MEG data. I look through the data and look for interictal epileptic activity, such as spikes or sharp waves, which are then used for analysis. Unfortunately, in this data, there is no such activity. We know that in about 20 to 30% of cases, we just don't record any spikes in the MEG during the limited recording time. In these cases, the MEG recording is therefore negative, and we're not able to contribute any additional information to the pre-surgical workup. And then sometimes we have these cases. This is a patient that seems to have multifocal epilepsy. In the MEG, we see activity over here in the temporal lobe on the right side. You also see activity on the left side, then even more on the right side again. This time it's perisylvian, maybe even close to the parietal and frontal areas. So it's basically all over the place. In this case, we also did not see any propagation phenomena, so we could not argue that it's starting at some point and then just propagating to some other area. And this is not limited to the MEG. This is an analysis, spike detection analysis of long-term video EEG monitoring, so many hours of data. And what we see here are also numerous spike patterns, again, all over the brain. On the left side and on the right side, perisylvian, temporal, parietal, frontal. Basically a very confusing, very complex scenario. Now, as you see, I put a question mark right there. So why did I do that? Because we know in this case that the patient is suffering from epilepsy because of this lesion right here. And this, again, just like in my last presentation, this is an MRI finding of a focal cortical dysplasia, also with a transmental sign right here. We know that FCDs are highly epileptogenic, so we know that this lesion corresponds to the epileptogenic zone. We also know that if we're able to completely resect this FCD, then the chances to get the patient seizure-free are really high. There's therefore no reason to believe, except for the interictal EEG, that this would be a multifocal case. So what can we do about this? The idea is now to find different markers in the MEG or EEG that might be correlated with the epilepsy and might also be useful for focus localization. So before I answer this question for the MEG, let us have a look at the invasive EEG. Because if there is something in the non-invasive MEG, some subtle activity, then we probably also should be able to detect that in invasive EEG due to the much higher signal-to-noise ratio. In a group of patients, we investigated the invasive EEG and looked for activity beyond the typical interictal epileptic discharges right here. Let me just show that right here. Indeed, there are other changes that occur, partially related to those spikes, partially independent.
Because maybe it's from
there is delta activity, more or less corresponding to what we see in the surface EEG and that we call slowing. Then we have theta activity, which might be just part of the spikes, but potentially also in other areas and other time segments independent of spikes. Then there's fast activity, like in the gamma frequency band, partially superimposed on spikes, but partially also preceding spikes, just like here. And then, of course, we have fast oscillations, high frequency oscillations, in this case in the so-called ripple band between 80 to 250 Hertz, which we know are highly specific for the epileptogenic zone. And here again, you also see them superimposed on spikes. And also preceding spikes like here and here. So the question that we asked ourselves is whether we would see a relationship between these markers and the quantity of those markers to the seizure onset zone in comparison to the irritative or propagation or unrelated areas. In addition to that, we were also interested whether this activity would also be related to the so-called dysmorphic neurons, which we know are the cellular correlates of the epileptic activity. These are some of the results. We compared electrode contacts in the seizure onset zone with contacts showing early propagation, as well as contacts in areas unrelated to the specific epilepsy. For delta, there is not much of a difference between onset, unrelated, and propagation areas, maybe just slightly elevated in the onset area, but nothing that we could use in these cases to determine the onset. Theta is more or less the same. Spikes obviously show a sharp contrast to unrelated areas. However, this is also due to the fact that we used findings of clinical routine to define the three zones in the first place. But then we saw a much higher amount of spikes, a much higher frequency of spikes in onset-related areas compared to areas that only show the propagation of the seizure. But this is not necessarily surprising. This is something that we could expect from the literature. And then we also see a very nice contrast in high-frequency oscillatory activity in the onset region. Much higher HFO quantities compared to propagation and basically nothing in unrelated areas. This is also something that is frequently reported nowadays in the literature. We also saw this contrast for somewhat slower fast activity in the gamma band. Here also you saw a high degree of gamma oscillations in onset areas, not so much in propagation areas, and not in unrelated areas. Furthermore, we evaluated phase amplitude coupling, a measure that evaluates the dynamics between the phase of a slow frequency, here for example theta, and the amplitude of a fast oscillation, here gamma. Phase amplitude coupling was also significantly increased in seizure onset zones compared to early propagation and unrelated areas. The question now, of course, is whether we can detect those also non-invasively using MEG or surface EEG. This is an example of how that might look like in a patient with frontal polar focal cortical dysplasia. I'm showing here the slow activity in the delta band as well as fast activity in the gamma band. On the very left, localization using connectivity is shown. In this patient, we could probably have used any of those markers to localize the lesion. The question, however, is now whether we can also find similar in other patients. Let's go through these different types of activity. Let me start with what I would consider something like the new black in epileptology, high-frequency oscillations. This is a case of a 43-year-old male patient with a right frontal focal cortical dysplasia. Successful surgery angle 1 outcome, and what you see here is a spike in just a few channels as an example. You see the spike pattern right here. Now, if you pay close attention, you see something else. Arguably, it's a bit hard to see if you have unfiltered data. Let me filter that for you. Here are the same channels, and what you see before the spike are high-frequency oscillations. In the time-frequency reconstruction, you see that this is in the ripple range, probably between 80 and 100 Hertz, so in the lower part of the ripple band. We found that regularly in this patient and used both the HFO and spikes for localization, which then yielded concordant results, which were also concordant with the resection. These are these results, and the focal cortical dysplasia is in this area. Now the question is whether what we see there really are to HFO's fast activity or high gamma, or whether these are just artifacts that you of course also often encounter in these frequency bands. Quite a while ago, back in 2010, we asked ourselves this question and we tried to contribute to the answer by recording invasive EEG and MEG simultaneously. This is an example what you see in such cases. Up here, you have unfiltered invasive EEG data, just a few seconds. You see spikes right here, right there. And then if you look closer, you also see the superimposed fast activity right here, which is more obvious, of course, when I filter this. Superimposed on spikes right here, then this burst-like pattern that's superimposed on those slower waves, but not necessarily a spike. And then this fast activity right here that has no real correlate in slower frequency bands, at least not related to a spike-like pattern. Now, this down here is the MEG data, but I'm showing you an average time frequency reconstruction over a number of channels over the region of interest. Here, you also see these high-frequency correlates in the high gamma band right here. Also a little less obvious here and here. To really reassure that what we are seeing is epilepsy-related fast activity, we use this for localization, and we're able to reproduce the resection area and the localizations of HFOs in the invasive EEG in almost all of these patients. So it seems to be possible to record at least high gamma, but how about faster activity? This is another example. This time we are recording ripples between 80 and 250 Hertz. This is MEG down here, and up here you see the simultaneous invasive recording. There is an MEG correlate to the invasive EEG high-frequency oscillations, but it's really hard to see. So if you now again take these MEG correlates and use that for localization, this is the result you get. This is the focal cortical dysplasia, and this right here is the invasive electrode that showed these high-frequency oscillations. This data shows again that there is high-frequency oscillatory activity in MEG. You can record it non-invasively and can also then use that for localization. But arguably, it's really difficult to do this without having a simultaneous invasive EEG, because already the detection is really hard if you just look at the raw data. This is our implementation of an automated fast activity detection in patients with epilepsy. These are just two examples of what that looks like. Again, patients with focal cortical dysplasias. Again, voxel-based morphometry showing the FCD right here. You see dipole localizations right here, and this is the corresponding localization of fast activity in the high gamma and ripples band. In this case, with FCD, the area with fast activity is more smeared, more extended. Now, this does not necessarily mean that this activity is really coming from these areas and that at least the irritative zone is as extended as this. It also might be an issue of source localization, because this, of course, is influenced by noise and the signal-to-noise ratio. So you would actually get larger areas if you have more noise. So always keep that in mind. Okay, let me now move to the other end of the spectrum, to what we call slow wave, corresponding to delta and theta frequency bands. Whether or not you can use the slow wave data for focus localization with MEG has been evaluated for quite some years now. This is a study by Ishibashi and colleagues. What they show is that by looking at such slow wave activity in the delta and the theta band, you can localize the epileptic focus just as well as using interictal spikes. In this study, that was the case in about half of their patients. So it might be an idea, if you don't have enough interictal spikes to perform source analysis, to also look at slow wave activity. This again is how that looks like in our lab now. This is yet again the patient with the frontal polar basal focal cortical dysplasia. On the right side, you see the conventional source localization using interictal epileptic discharges. On the left, from the same dataset, you get this delta localization. The idea is that you calculate how much delta is present at each point in the brain using a 3D grid with a specific resolution. You then compare this to a database of healthy controls to see where the patient exceeds what you would expect under normal circumstances, or healthy circumstances at least. Right here, you see an increase, especially in this area, very close to the focal cortical dysplasia. But this also extends to more rostral areas, and it's more diffuse, which is exactly what we saw in histology and also in invasive EEG. Remember this case, the so-called multifocal case, where we had focal cortical dysplasia in this area, but saw interictal spiking activity all over the place in MEG and EEG. We also calculated slow wave analysis in this patient, again from the same data that we also used for this spike analysis. And these are the results shown here. Again, his MRI with the focal cortical dysplasia, and this is the slow wave or delta localization. As you can see, the significant increase is only located in the area of the focal cortical dysplasia. There's nothing on the other side, nothing in other lobes. This is very unlike what we saw using interictal spikes. Since this is from the same dataset, that also seems to suggest that this slow wave or delta activity is not necessarily the same thing as these spikes. Otherwise, you could argue that you might just be seeing the wave component of a spike wave complex. If that would have been the case, then you would have also seen some of the slow activity on the other side, where we definitely saw interictal spiking. These are some preliminary results on the diagnostic accuracy of slow wave localization in 50 patients with different etiologies, ranging from FCDs to hippocampal sclerosis, tumors, and others. We compared our finding in slow wave activity with the consensus of pre-surgical evaluation, so all the other methods of pre-surgical workup, that is MRI, video EEG, invasive EEG, PET, SPECT, and so on, and also MEG using conventional analysis. On a regional scale, you see that there's quite some overlap. First of all, significant delta increase above what you would expect in healthy controls was observed in 34 patients, so roughly about 70%. In 29 of these 34 patients, the finding was concordant or overlapping with the consensus of pre-surgical evaluation. In 5 of these 29 patients, we had no or very few spikes, certainly too few spikes for robust source localization using conventional means. Let me now get to the last part, using connectivity for focus localization. The idea here is that the brain is a complex system of networks with dynamic communication between different hubs. In epilepsy, the focus would be such a hub which shows a lot of spontaneous activity that might be interictal spikes, seizures, or subtle interictal oscillations. This activity, however, does not stay confined to just the focus hub, but is communicated to other parts of the network system using pre-existing or altered connectivity pathways. Then it might be possible to find this focus hub by investigating the ongoing communication statistically. This is how connectivity localization looks like in our lab. Again, I'm using patient JD as an example. As you can see, we consistently see connectivity increases which are nicely concordant with the frontopolar focal cortical dysplasia. This finding was largely independent of the chosen frequency band. Broadband, alpha, delta are shown as examples, which all show a maximum coinciding with the FCD. In a recent study, we evaluated the concordance of those connectivity increases and seizure onset zones from invasive EEG in 50 patients. The results on the left show that especially in the delta, theta, and beta frequency band, such maxima can be used for localization, with median area under the curve values just below 0.8. However, there's some variability and the choice of frequency band is not entirely clear, so there's certainly work to do before we can use this for clinical practice. So summing up, there is increasing evidence that electrophysiological correlates of epilepsy are not limited to spikes, sharp waves, and seizure activity. Above and beyond those patterns, there seem to be oscillatory changes which may be too subtle for conventional visual analysis, ranging from slow wave to gamma, high-frequency oscillations, and connectivity. Current data shows promising results on the additional clinical value of such markers, especially in unclear cases with no or not enough spikes on the one hand, or very diffuse and multifocal findings on the other may benefit from these methods. They are also interesting from a practical perspective, as the procedures are largely automated, so applying them should be straightforward and not very time-consuming. However, all of this is rather new. Correspondingly, we still need to do much work on validation and on the optimal methodology. Thank you very much for your attention.
All right, that was a very interesting presentation by Dr. Stefan, Rampp. I don't know how tight we are, Arioski, time-wise, so I know we are running over time based on the schedule, but do we have a bit of time for questions?
Yeah, my question is for you concerning the different clinical profiles of Alzheimer's disease patients. So, for example, early onset patients with Alzheimer's disease may have no very, very important episodic memory deficits. So do you have some results on early onset Alzheimer's disease patients with your approach?
Yeah, well, yes, we do have, especially in the genetic variants, right? So I haven't studied yet a patient with late onset sporadic Alzheimer's disease. So we're talking about So then, but in early onset familial Alzheimer's disease, we have investigated substantially this population, especially those who carry the gene mutation that I alluded to during my presentation, Claudio. We have now detected the earliest memory impairment, which are not episodic. These memory impairment are working memory impairments in the binding ability that I shared during my presentation, which appear before episodic memory deficit becomes apparent. We have now some understanding why this is the case, why these low-level memory, integrative memory abilities are targeted by the disease before episodic memory stages where the hippocampus is affected, which we now know is not in the earliest stages of Alzheimer's disease. But we have also seen, Claudio, which I think is relevant to this meeting, is that those memory impairments that are present in this genetic population in the pre-symptomatic stages and early symptomatic stages are characterized by an increase in brain activity. So in fact, we published in Alzheimer's Research and Therapy a few years ago a study that we showed that over hyperconnectivity over centroparietal regions classify with over 90% or 89, to be more precise, percent accuracy, asymptomatic carrier from non-carriers. So high reduction in connectivity was only seen in one of these carriers, and it was in more advanced stages of the disease. Again, indicating that that was the point I was trying to make during my presentation. We need to look at the EEG along the continuum, envisaging that there will be fluctuations, and this fluctuation, meaning increases and increases, will be contingent upon where people are in the disease continuum, and will be telling us potentially interesting story about the likelihood of progressing to dementia. As Paolo said earlier, as a standalone tool, probably is not going to be a great diagnostic tool, but combined with other methods, we can grant potential specificity to the EEG observations that we are drawing from this study.
Hello, everybody. I just want to sort of squeeze in there. This is a really fascinating sort of discussion. I just note that we are 18 minutes over time. Apologies for this. Really fascinating sort of discussions. I don't want to sort of interrupt and stop that flow, but unfortunately, we are going to have to sign off for today. Good news is there's more. There's more time tomorrow. Of course, we have the final day of the GBC event. So please do keep these conversations going, perhaps even over email in the meantime, or perhaps tomorrow when you our plenary session as well. So just a couple of very quick things to sort of say. First of all, is to thank you all, the speakers and sort of the delegates for listening in. And if anybody would like to get an attendance sort of certificate, you can email the GBC address, which has been posted in the general chat, and you can reach out and get that a couple of days post event. And finally, we'll of course make all of the videos available. Those that you couldn't make because you were in this session, you'll also get to see the full program eventually.
That's fantastic.
For that. So thanks again, everyone. See you all tomorrow, hopefully. Okay. So all the best. Thank you all.
All the best. Thank you for the support. Bye.